Greetings. It's wonderful to see all of you again today. Thank you for joining us for another one of our series of town hall conversations. This is actually now the 28th of these conversations, and I'm very delighted, as always, to thank the members of our Society for Leading Medicine, the chairs of our Society Leading for Medicine, Jeremy and Caroline Finkelstein and Catherine and George Masterson, for their leadership. And it's wonderful to see all of you again today. We did our very first one of these, now it's 28, back in April of 2020. And of course, now we've morphed them really mostly away from COVID, some small updates and focusing on other great things that are happening at Houston Methodist. But that first one was a time of great uncertainty and a time where we were focusing on what we were calling then not COVID-19, but a novel coronavirus, something that had followed on the heels of SARS and MERS, which were two previous severe coronaviruses that we had dealt with. And actually, Dr. Jim Musser was one of the guests then and has gone 26 in between, I guess, without being here, and is joining us today. We've learned an awful lot in those 2.5 years, and one of the things that was really underscored was the need for us to lean in, and as we've led throughout the COVID pandemic as an institution, really be there and push infectious disease as one of our key core centers of excellence. And Dr. Musser has been at the center of doing all of that during this time, and he's going to tell you a lot about that today. Today we also have Dr. William Miller in the background texting furiously, so bring in your questions there through the chat line, and he'll be happy to answer your questions. So I'm going to turn things over now to Dr. Jim Musser. Dr. Musser is the chair of our Department of Pathology and Genomic Medicine. He also holds the Fondren Presidential Distinguished Chair, and he's going to introduce our other colleagues here today who are also going to share a lot of our work in infectious disease. Dr. Musser.
Thank you very much, Dr. Boom. It's a great pleasure to be here this morning with the group. A brief introduction of my two colleagues who are here with us today from the infectious disease laboratory. First individual is Dr. Cesar Arias, who is my colleague and serves as the co-director of our new Center for Infectious Diseases. And then also here with me is Dr. Jimmy Gallagher, who is a addition to the group January 1 of this year, and he runs what we call the ADAPT laboratory, and you'll hear more about that later on in the presentation. So let me just spend some time now reviewing the last few, it seems like 15 years, but of course it's only been about 2.5 years of our response to the SARS-CoV-2 pandemic. And then at the end, I'll provide an overview of the birth of our new Center for Infectious Diseases. So I think a key take-home message is listed here. In the Department of Pathology and Genomic Medicine, we have used the devastating SARS-CoV-2 COVID-19 pandemic to hone our strategies for subsequent pandemics, regardless of what they are or when they occur. And many folks now call this the disease X scenario because, simply put, we never know what's going to be the next one. In the Department of Pathology and Genomic Medicine, almost from day one, we put into play a four-pronged strategy. One was to do rapid molecular diagnostics. The second one was to provide rapid virus genome sequencing. We also put into play the antibody therapy, the convalescent plasma therapy that you've heard a lot about, and then antibody testing as well. These four areas were led by members of the Department of Pathology and Genomic Medicine. This was a highly integrated effort. Dr. Randy Olson leading the molecular diagnostics and genomics strategy. Wesley Long doing whole genome sequencing and analysis. Eric Salazar spearheaded the convalescent plasma therapy project. And then Paul Christensen, a very talented informatics individual, who led our clinical and research informatics and the important aspect of data integration. Importantly, we have always in the department developed molecular tests for problematic pathogens. I've just listed some of these here, really dating back to 2009 when we rapidly put into play molecular testing for, at that time, the H1N1 problem. And then you see some of the others listed on the slide as well. The rapid development and deployment of our molecular diagnostics tests have really taught us rapidly how to formulate the same strategy for a SARS-CoV-2 pandemic. This is a very busy slide. I just want to show you that there's been a lot of activity since December 31st of 2019, when the first case cluster of pneumonia was presented or recognized in Wuhan. We rapidly, beginning in January, began to investigate our options for molecular testing. And I'm very pleased that by Valentine's Day that year of 2020, we already had our test validated and ready to go before the first case was diagnosed here in Houston. And then you see up now through September 22nd, last week, we've done well over 1 million SARS-CoV-2 PCR testings performed. So it's been quite a busy time since that first one. Here is a overview of the 6 waves that we've seen in Houston and we've experienced. You see the big Omicron wave, the 5th wave occurring caused by the BA1, BA2 variant, and now we're into the 6th wave that really has now caused essentially all of our cases are BA5 in this 6th wave. Here's just a few statistics. I mentioned the 1.2 million PCR tests performed. Let's skip down to the bottom there where I'm telling you that we have also sequenced the genome of 115,000 SARS-CoV-2 infected individuals in the Houston Methodist system since that time. So very, very busy time. Importantly, we are uniquely poised in the Methodist system, hospital system, and healthcare system to really, in a seamless fashion, throughout the 7.2 million population in the Houston area, we are uniquely poised to have an outstanding opportunity for unique infectious disease preparedness, response, and research in this very large metropolitan area here in Texas. Our integrated activities and their analyses have led the nation in many areas and garnered extensive national and international attention. And I'll just share with you 3 examples of this. One was the analysis early on of greater than 5,000 Houston area SARS-CoV-2 strains. This was initially a very big effort, and we discovered several things, including what had happened was this massive genetic study, and this was picked up by the Washington Post as one example. It really showed that the coronavirus was mutating and evolving amid rapid US spread. So that garnered a lot of national and international attention. A second area that also got a lot of national attention was Eric Salazar leading our convalescent plasma treatment. We were able to demonstrate there was a significantly decreased mortality in our large cohort of COVID-19 patients that were transfused early, and that's crucial to transfuse early with high titer, what we call high titer or potent convalescent plasma with anti-SARS-CoV-2 spike protein. And we really we were the first in the nation actually, late on a Saturday night, almost a Sunday morning, to infuse a patient with convalescent plasma. And then a third area or third publication that we were leading on was when Omicron arrived. We rapidly reported there were signals of the significantly increased vaccine breakthrough, which was quite common at the time with Omicron, and unfortunately decreased hospitalization rates and less severe disease in patients who experienced the Omicron variant of SARS-CoV-2. So those were just 3 of the contributions that we've made over the last 2.5 years. There are 2 very important reasons that we were that things worked well for us here at Houston Methodist. There was the rapid, clear, unambiguous, and unwavering support from Dr. Boom and all Methodist leadership. The second thing that was really important to point out is there was an extraordinarily generous community support literally from day one. And really, without these 2 points that I've made here, very little in the arena that we work in would have been accomplished. So it was really the unwavering support from Methodist leadership and the extraordinarily generous community support that permitted us to really have great effect here on the SARS-CoV-2 problem. A few lessons that we've learned, and this is the very, very, very short list. I'm just pointing out a few things here. The great majority of pieces to the puzzle were under one roof, that is one Methodist culture roof, and they were seamlessly integrated. And I can't stress enough how important the seamless integration was to our success. The flat administrative structure here at Houston Methodist was absolutely essential. Decisions were made very, very rapidly, very early on. I remember having conversations in the hallway with Dr. Boom, and he simply said, yes, go ahead and do whatever I was asking him if we could go ahead and do. So that was really crucial, rapid decision making. And this really, the rapid decision making coupled with the philanthropic community coming together to provide substantial resources that permitted us to really move forward and attack the pandemic rapidly. So this has led, as Dr. Boom alluded to in his opening remarks, to the birth of the Center of Excellence in Infectious Diseases. We recognized very early on in the pandemic that we had several limitations and deficiencies that we needed to address and gaps to fill. So the first one we realized is that we were not seeing what unfortunately would be the last pandemic. There will be additional ones in the future because of many reasons: the globalization that we see, the urbanization, bioterrorism is always in the back of our minds. We've got the rogue nation state problem and susceptibility and the ease of genetic manipulation of pathogens. So these are reasons that we are absolutely certain we really must continue to be vigilant in the future and expand our capability here. The key goal, one of the key goals of our Center for Excellence in Infectious Diseases is to be exceptionally well prepared and equipped and have the ability to address the new threat extremely rapidly. Extremely rapidly means within days. Remember that certainly with Omicron, those cases were doubling every 3 days. So the more time lag that occurs before confronting the new problem, the worse off we are going to be. And we've got to do this to fill these gaps, all the while continuing to focus on our common pathogen problems such as MRSA, MRSA, other drug-resistant bacteria, sepsis, and so on. So to address initially some of our gaps, we first recruited a world-class co-director who is a visionary infectious disease physician, scientist, and outstanding clinician. One individual in our search, we did a national and international search, one individual was head and shoulders above all the other candidates, and we were very fortunate to attract Dr. Cesar Arias to our Center for Infectious Diseases and to our Department of Medicine to be the new chief of the Division of Infectious Diseases in the Department of Medicine. And you'll hear more from him in just a bit. We also needed to fill other critical research portfolio gaps. One was we needed antibody discovery and therapy development platform on site. We needed it to be agile, flexible, very high throughput for rapid, and I stress rapid, therapeutic human monoclonal antibody discovery using these novel and really interesting strategies called synthetic biology, artificial intelligence, and protein engineering. And you'll hear more about that in a bit from Dr. Jimmy Gallagher, who is sitting to my left here. He is our new head of the Laboratory of Antibody Discovery and Accelerated Protein Therapeutics. We call it the ADAPT Laboratory. He has a tremendous background in this area. Previously, he was the chief technology officer for an entity called Biomade that was part of the Army Research Laboratory. He was chief technology officer of synthetic biology and the Essential Research Program. And he and I were fortunate to meet each other 32 months ago and develop extensive collaborations. And then this led to our ability to attract him here to head up the ADAPT laboratory. And as I say, you'll hear more about this fascinating strategy later on in the town hall. So we have additional portfolio gaps to fill. We currently have ongoing recruitments in several areas. One is to bring on board and recruit and successfully bring on board a human virologist, a human immunologist to focus on human infection immunology, and then an RNA vaccinology individual. And this will permit us to leverage our outstanding expertise that we already have here in RNA biology, led by Dr. John Cook's RNA group in the Research Institute. So I'll close there. Thanks everyone for joining in, and I'll turn over the pointer and baton to my colleague, Dr. Cesar Arias.
Thank you very much, Dr. Musser and Dr. Boom, for this kind invitation. I represent here the Department of Medicine, and I serve as Chief of the Division of Infectious Diseases as well. And I'm going to try to give you a perspective from the trenches, since we have been dealing with all these infectious disease threats currently. So I have some disclosures that come to the institution as grant support. So this is what I'm, in this few minutes, this is what I want to discuss. We want to talk about a little bit about the COVID pandemic and these updates in vaccination. We're going to talk about the monkeypox outbreak that we are all experiencing at this moment. Polio in the United States, that is a new re-emergent threat. And then delineate another infectious diseases threats. As I say to my trainees, residents, and other colleagues of mine, infectious diseases is one of the most exciting branches of medicine because we all have a new disease almost every year. And we had experienced that, unfortunately, in the last couple of years and more. So this slide is to show you that we're still in a phase of the pandemic in which we have still a significant number of cases that we have daily in the United States. More than 50,000 cases are still being recorded in the United States, with about 400 deaths in the United States. Hospitalization declining, but most importantly, as you can see in the right part of the graph, so although the vaccination of initial doses have increased, the vaccination for boosters continue to be lagging significantly in our population. And that produces an important vulnerability for our most susceptible population. This is data from the CDC and the FDA in a recent call that we gave with the Infectious Disease Society of America. As you can see, as Dr. Musser showed, the different waves, but I just want you to concentrate on the right part of the graph, where since April, hospitalization rates in older age have increased relative to other age groups, and those are the people that are more vulnerable. And one of the main reasons for that is because the type, as Dr. Musser pointed out, the evolution of this virus has changed significantly. And this is just data showing you in different colors what sort of variant are dominating now in our population. And that green bars, as we move towards now, September 2022, is dominated by those Omicron subvariants called BA4, particularly BA5 at the moment. And those colors represented in that column signify what are the dominant strains. So this virus has evolved significantly, and this is not unusual from coronavirus, which produces several challenges. And one of the main challenges of this evolution of these viruses is that the immune responses that are mounted against original variants can change significantly. And because of that data that I show you, that more vulnerable people are more hospitalization increasing in the last few months. That was this particular input of impetus to eventually produce new boosters. And this is summarized in this slide. So on the left panel, you see what happens with the immunity. In this case, we're only measuring neutralizing antibodies before booster and after the booster. And as you can see, the colors on the left side with the new variants, particularly BA4 and BA5, before the booster, there is no detectable neutralizing antibodies. And after boosters, that response, or the antibodies that are detected against those viruses, those particular variants, increases significantly from 3 to 14 times, which is sort of a surrogate of some sort of protection. And on the panel on the right, my pointer is just, okay, now it's sort of, in the panel on the right, as you can see here, this is the infection rates, patients that are naturally infected with these variants, BA1, BA2, and other subvariants. When you have sort of more closest virus, then you have antibodies from natural immunity that confirms that is the exposure to these viruses or the antigens of these viruses that potentially give you some protection. So because of that, people who were vaccinated but not boosted seems to be unprotected. And putting together this data with the data of increased hospitalizations in the older population that are the most vulnerable is what drove the decision by the CDC, FDA, and the government to recommend these new boosters. So this is exemplified here of what is happening. So as the new novel variants emerge and increase in frequency in our population on the left, then waning immunity in the population, that is increased risk of major COVID outbreaks. And on the right panel, you see now the CDC has a very excellent team of modelers or how, based on different data that is input almost daily, what's going to happen with the trends. And the graph basically shows here from July to the end of the graph will be October, November. And what the graph shows that there is a possibility that we will have an increase of infections starting in the fall and peaking in November, December. So with this prediction of the fall being is vulnerable timing, that also endorsed the decision to try to protect people as much as we can with these new boosters. Okay, so the current vaccine recommendation now is that the Moderna bivalent COVID vaccine plus the booster we call the bivalent was authorized in ages 18 and older. The ones Pfizer bivalent also from 12 and older. The primary series dose continues with monovalent Moderna and Pfizer vaccine as the original vaccine, but the boosters that were applied before are no longer valid, so they have been revoked. And if anyone wants to boost, needs to be boosted with these bivalent boosters, which includes these new variants for obvious reasons and the reasons I just explained to you. So these are sort of the most common recommendations. I'm going to stop there with that. I'm going to talk a little bit about monkeypox. This is another disease outbreak that we are facing, and as infectious disease clinicians, we've been able to see several patients in our system. This is a disease that was endemic in Africa and now has spread around the world. This is about 3 days before this slide. We have now more than 25,000 cases in the United States. This presented with some unusual rash in different parts of the body. But one of the things that we see when in the front lines is these presentations have been very different from what was described before, particularly attacking genitals, producing several problems, oral and genital lesions that are very difficult to treat sometimes and very distressing for the patients that are suffering this. This is a map that shows in yellow what is the non-endemic monkeypox epidemic. The size of the circle is the number of cases. As you can see, the United States is leading the number of cases around the world, mostly because we have very good ability to diagnose it, but it is all over the world right now, with more than 65,000 cases now reported from this outbreak. The transmission of this virus is person-to-person, direct contact, basically, that can do in any activity, or human activity, with respiratory secretion, touching lesions, and some clothing and items of patients that have these lesions that are teeming with viruses. And the patients are infectious from the symptoms that they infect until the lesions basically scab, and there is a fresh layer of skin forms. I know this outbreak has been reported in a certain group, and this is a sexual networks that have increased the transmission of this virus, but anyone, including children and pregnant women, can be susceptible to this virus. So we have a vaccine, contrary to COVID-19, we have a vaccine we call the Genius vaccine. There has been more than 461 patients that have received vaccines, both as pre-exposure prophylaxis to protect patients and laboratory personnel and healthcare workers, and post-exposure prophylaxis to decrease that sort of effect of the disease, and is being deployed around the United States. And we also have treatment that we don't have a lot of clinical data. It's a clinical trial in progress, but our clinical experience with this drug is very positive, and that can curtail the symptoms and help patients get better. The drug is called TPOXX, that's been in the press for a long time now, and has both oral and IV formulations of that. Finally, two things that I wanted to mention particularly. One is that these infectious diseases don't stop coming. With the global warming, the encroaching of the population in wild areas, we continue to have things. The latest disease about a month ago, the report of polio in New York. This is associated with the vaccine, and what happens here is people don't get vaccinated, the level of immunity decreases, and some of these strains, particularly given that outside of the United States through oral administration for vaccine, can potentially mutate and produce eventually polio, which is a neurological devastating disease that can cripple people. So we have this in the United States, and as you see there in the vaccine, the poliovirus is likely to be continued transmitted in this county in New York, and there's several investigations now going on with this. And I want to forget, one of the reasons I came to Houston Methodist is because the silent pandemic I call antibiotic resistant is important. This is projections from the UK government that in 2050, it is likely that antibiotic resistant organisms will kill more people than cancer. And that has given a lot of impetus to research on that. And this is just recent data estimated that approximately 5 million people died in 2019 of antimicrobial resistant organisms. So this is something that is not going to go away, that we see, particularly Houston Methodist, with all the complexity of our patients on the daily basis. Finally, the last virus that has been in the press is this virus called Henipa virus that was described in China, that causes a febrile illness. Basically, is a relative of viruses that have been described in India, like Nipah viruses, but it is an intermediate host, is the shrews, and this is an important situation where zoonotic in contact with animals that we probably do not respect their habitat as we should, can produce new outbreaks and new virus getting into human beings. So our job doesn't stop. It's going to continue forever, kind of job security, but at the same time, threats that we need to be prepared. And we are putting everything here in Houston Methodist to be prepared for all these challenges. Thank you very much for your attention.
Great. So now we'll hear from Dr. Jimmy Gallagher. Jimmy, in addition to all his accomplishments, importantly, is a native Texan, one of the rarities, at least in my experience. But he's a native Texan. So Jimmy, the floor is yours.
Thank you. I wanted to talk a little bit about the research that we do to complement what you've heard from Drs. Musser and Arias. So my laboratory, ADAPT, or the Antibody Discovery and Accelerated Protein Therapeutics Laboratory, really focuses on ultra-high throughput discovery of therapeutics to diverse pathogens or diseases. I want to really emphasize that while we'll be talking a lot about COVID today, everything that I tell you can be applied to just about any disease that you can think of, from infectious disease to cancer. So my group really sits at this intersection of systems immunology, synthetic biology, and machine learning, and we use these tools to engineer monoclonal antibodies, vaccine candidates, enzymes, and just about anything that is made out of amino acids, which eventually become proteins. We have a really interesting technology portfolio that really kind of encompasses everything in engineering biology, from artificial intelligence and machine learning to computational protein design and genetic circuitry engineering to high throughput screening and selections, and then also developing turnkey automation pipelines so that we can rapidly respond and proactively create therapeutic candidates. I am joined by an all-star cast of scientists and engineers that span the expertise needed for biological engineering. You heard a little bit about me earlier. I'll now turn your attention to my team. So Dr. Raghav Shroff is a protein engineer and artificial intelligence model builder. In the last month or so, you probably heard about a plastic degrading enzyme that was engineered using Dr. Shroff's machine learning algorithm. It could take plastic and degrade that back into monomers for recycling. We're now using his algorithms to improve therapeutics. We also have Dr. Dan Bouts, who's a pioneer in systems immunology and monoclonal antibody identification in serum. We also have Dr. Andrew Horton, who is a systems biologist and computational guru. Dr. Thomas Segal Shapiro is a bona fide synthetic biologist, helps out a lot with our genetic circuitry design and our laboratory automation. We have Dr. Yula Goika, who is an antibody expert, and Dr. Shanak Carr, who helps us engineer enzymes and is also a synthetic biology expert. Of course, we're all corralled by our laboratory supervisor, Michelle Byram. So now I'm going to move into a couple of examples of what I mean by platform technologies. And the first one is what we call spike display. So we wanted to come up with a platform that would allow us to test spike proteins as they were evolving. So as we all know, SARS-CoV-2 is the causative agent of COVID-19, and the business and target really is the spike protein that you've heard so much about. The spike protein sits on the surface of the virus and can attach to our cells through an interaction with the spike protein to the ACE2 receptor. This causes it to shapeshift and allows the virus to come into the cells and replicate. So this is the target for therapeutics, and if you've had a US approved vaccine, you've had a pre-fusion stabilized version of this protein. What we wanted to ask was, is there a way to rapidly build new variants as they emerge and test monoclonal antibody treatments that are already approved or ones that we were developing. So we developed what we called spike display, which allows us to put variants on the surface of the mammalian cells in a very safe manner. We don't have to make virus, we don't have to culture virus, and it allows us to test antibody binding, expression, and the ability of the virus to attach to the ACE2 receptor. Our first proof of concept with this, we used 8 variants of concern using the Wuhan all the way through BA1 and BA2, the Omicron variants that you've heard so much about. And when you see red in the box below, that means that particular class of antibody does not bind. And as you can see, when we got down to Omicron, most of the therapeutic antibodies that were known were completely escaped. We corroborated these results with live virus neutralization with our partners at UTMB, and that's what you see on the right-hand side of the screen. So this gave us confidence that we'd built a platform that could recapitulate live virus neutralization in a safe laboratory environment. Now, Omicron had an unprecedented number of mutations, 29 and 33. Some of them were shared between the 2 variants, and we wanted to ask the question, can we figure out how these mutations evade host immunity in antibody treatment? But of course, that would be very difficult to do by hand and one at a time. As you can see, we had to build over 200 genetic constructs to put into our pipeline of this design, build, test, characterize framework. So we built computer software that allowed us to take sequences and variants and automatically generate lists to synthesize, to test, that we could then tell our laboratory automation how to stitch these together so that we could test them in the lab. We've very recently been able to link this up to our in-house sequencing efforts that you heard Dr. Musser tell you about earlier. The take-home message is that we were able to identify the reason or the cause for evasion in very fine molecular detail, and we also uncovered new mechanisms of host evasion. So it appears that Omicron was not only mutating individual amino acids at sites where antibodies bind, but was also changing the dynamics of the protein, making really potent quaternary binders also less effective. Now, in the future, so that we can prepare for disease X, we need to increase our throughput from hundreds to tens of thousands to millions. We want to expand this platform to other emerging viruses, and then we want to use this platform in an engineering strategy to engineer pre-fusion stabilized proteins to different viruses. That way, when disease X comes, we're not responding, but we're pulling it out of the freezer. Of course, we work with the Center for RNA Therapeutics. Not only do we provide candidates for vaccines, but we also engineer components to make mRNA manufacturing easier and more scalable. Next, I'm going to briefly touch upon rapid response biological countermeasures. So this is our antibody discovery platform, and if you're not an immunologist, that's okay. The important message here is that antibodies are what protect you, and these are one of the fastest growing classes of drugs. Convalescent plasma is sort of the polyclonal version of this, and so what we try to do is go in and find monoclonals that are broadly protective and very potent. So we do this a little differently than traditional antibody discovery platforms. So we use a semi-synthetic approach where we will take natural repertoires from people and combinatorially combine and test them to screen for the best possible combinations, because not all antibodies that we develop in our immune system are the best that they could be. And in fact, most of the time, they're just good enough to get by. And so we're developing platforms to really speed not only the discovery, but the optimal engineering of monoclonals. Just as an example, so We can do this very rapidly. During the current SARS-CoV-2 pandemic, we went from patient samples to validated antibodies in just under 4 months. We actually had our first candidates in weeks. However, we didn't have the testing infrastructure with live virus neutralization until a little bit later. We've learned quite a few lessons since starting this platform, and we are now to the point where we are bottlenecked by GMP manufacturing and being able to scale up antibodies. So you hear a lot about monoclonals being discovered. There was a time where every month there was a new potent neutralizing antibody. What you didn't hear is that the scale up and the manufacturing of these antibodies is actually quite difficult. So we are attempting to build platforms that have manufacturability up front so that when we go to scale up, we are able to make antibodies that can be used as therapeutics. We also use artificial intelligence, machine learning. We do this in a variety of use cases. One is to develop mRNA vaccine manufacturing enzymes. 2, identify scalable monoclonal antibodies. We want to enhance the properties of medically relevant proteins, and we want to develop models to improve the speed of biologics engineering. Of course, this is all made possible because we are at Houston Methodist Hospital. The Research Institute was put next to the hospital for a reason, for translational activities, such as those that happened during the pandemic, and we can get to phase 2 under one roof. So that's a little bit about the ADAPT laboratory. Thank you for your time and attention.
Well, thank you very much to the whole team here. It's a fascinating presentation, as I always see here. I learn a whole lot every time I get a chance to talk to all these amazing people that we have at Houston Methodist. So we have a nice amount of time today for Q&A, and we've been getting a fair amount of questions coming in over the wires. Please keep them coming if you do have other questions. You know, I guess I'll knock out an easy one that we get asked all the time. Can you get the flu shot? I'm going to ask with Dr. Arias, can you get the flu shot and the COVID booster at the same time?
Yes, absolutely yes.
Yes.
So the only caveat for that, for example, I spaced it for a week just to make sure if you have side effects, you may not double down. But you know, I know many people who have both at the same times, and there is no problem to do it. Right. And I didn't mention we are experiencing, you know, a higher activity of flu at the moment, and I strongly recommend everybody needs to put in their flu shots.
Thank you. So yeah, it's so. Really, the bottom line is our immune systems are pretty darn sophisticated, right?
Absolutely.
So they can handle a couple things coming at them at the same time because they're getting tons and tons of things normally coming at them at the same time, just sitting here and interacting in life. So
Absolutely.
They can mount an antibody response to flu. They can mount an antibody response.
And then we get protected, which is the main target of all this.
So drill down a little more on your comment about flu. That was one of the notes I'd made. And actually, I apologize. I'm going to turn that to Dr. Musser because I think one of the really cool things we have available on our website is a flu tracker. And if you pull up our flu tracker, it doesn't look like it has looked in previous years right now at the end of September.
Yeah, that's correct, Dr. Boom. So what Dr. Boom is alluding to is that we post all of our data publicly dealing with respiratory pathogens. It's all available on the Methodist Flu Tracker. And what we're seeing, unfortunately, is very early influenza A disease this year. It generally is much later in the year and sometimes into the new year. But this year, we're actually seeing an uptick in flu A activity that began earlier in September. We're very concerned about this. And just to reemphasize what Dr. Arias says, please get your flu vaccination as well. We really want to avoid what many have alluded to as a twindemic, that is both flu and COVID in the circulating and causing a lot of disease at the same time. So you're welcome to visit the website. It has all the data for our respiratory pathogens, and it'll show a pretty substantial uptick early on this year, which some have ascribed to the fact that in the southern hemisphere, there was also a lot of flu activity, and traditionally what occurs in the southern hemisphere translates into what is going to occur in the northern hemisphere, and of course here in Houston, Texas as well, and that's exactly what's transpiring.
Great, thank you. You know, as a primary care physician, I'll often, in many other years, kind of working with patients I know are very reliable, they're going to 100% get their flu shot, say, you know, and my wife and I do this, you might time it a little later in October, kind of hit it peaking, make sure on the tail end you're in good shape. My patients, I'm not so sure going to get it. I'm just like, get it now. But this year, I'm telling everybody, get it now. And in fact, under here, I've got my Band-Aid from yesterday. 'Cause I got mine yesterday, and strongly recommend you do go ahead and get your flu shots as well. So, you know, one of the complexities now with this set of COVID vaccines and this booster is, you know, we've had so much breakthrough. So everybody's got their own sort of individual timing and story. And some people qualified for one booster, some people were qualifying for 2 boosters, some people haven't gotten any booster, some people got the booster and then got sick, you know, I mean, so there's all sorts of timings. Could you kind of try and break that down? I have one specific question here from somebody who, for example, got 4 vaccines, so 2 boosters, the last one in mid-May. Then in July, because we're having breakthrough infections, it's protecting against severe disease, but we're having breakthrough infections, had COVID. When should they get this booster? And then I guess even a little more provocative, why should they get this booster?
Yeah, those are very good questions. I actually have 5 boosters now, plus COVID. So one of the clear situations here is that, as I mentioned to you, the viral evolution versus the protective immunity seems to be in a crossroads. So as more time goes after your boosters, there is a decline in your protection, and then that allows for some variants to emerge that evade those. And that seems to happen with these vaccines between 4 and 6 months, when you start seeing really a good decline. So in general, it's recommended that people who have had a booster more than 4 to 6 months, they should consider now having their boosters done, because now we have a new variant. And that's actually very important to time that. If you got COVID, it's like you had a booster. And of course, depending where you had COVID, when I had COVID in April, was not BA4, BA5, was probably BA2, which actually protects me a little bit against BA4 and BA5. But since I am traveling now and I'm going to visit my parents in December, I got boosted. So the recommendation now is minimum of 2 months. I would say once you have a booster, you probably, I think probably the ideal time is about 4 months, 4 to 6 months to get boosted. And it's probably very likely that we will need boosters on a yearly basis, since I don't think this is going to go away.
So one of the things we've clearly seen, I think some of this obviously is viral mutation. Another issue is really the issue of a respiratory virus kind of getting in and breaching our defenses, right? We're still kind of getting the army of antibodies and T cells and everything kind of getting revved up to prevent severe illness, but we've had all these breakthroughs. Is there, I guess, explain a little bit why that is, kind of those initial line of defenses, and then is there research happening in different types of vaccines, different ways we might address that might give us a little more effective, durable sort of frontline defenses in our upper respiratory tract?
Yeah, definitely. That's sort of the first line of defense. And just to answer probably the second question, there is now interesting data with inhaled vaccines to try to elicit a sort of mucosal immunity, and that will be easy to administer and just do it when you do. Going back to your first point, I think it's very important to realize that mucosal immunity is important. And as you see the data, these vaccines have prevented severe disease and death dramatically. And that saved millions of lives. But not only that, I think now we have pretty good data to suggest that also prevents transmission. So it's not only you, it's your relatives. My 86-year-old mother who has COPD and cardiovascular disease who is at risk of having a bad disease. If I get vaccinated, she gets vaccinated, the odds that that's going to be a bad outcome decreases. So that mucosal first entry seems to be very critical to handle this disease.
Well, thank you. Thank you. There's questions oftentimes come up about, can you get too many boosters? And is there a negative impact perhaps on the ability to mount an immune response with more boosters? Can you talk a little bit about that?
Yeah, so there are some data, for example, in pneumococcal vaccine, where there are two different pneumococcal vaccines. If you put them together, one vaccine can potentially alter the response of the second one. So, people sort of try to spread them out in a year, and that's sort of the current recommendations. In terms of boosters, since the doses are less and then they are targeting sort of the same target, it's unclear that if you do it too close, that probably happens. But then with the time frame that we mentioned, there seems to be a response, particularly when the booster is directed from a circulating variant that was not protected by the previous vaccines. Again, the original vaccine seems to be protecting very good. And then you are young, healthy, and all that. It's good to think about it. Do I really need it? But if you have eventually exposing your parents or exposing other people you have to take that into consideration. And everyone at this point should weigh that risk.
Great. Well, that's very, very, very helpful. Thank you. Well, Dr. Musser, I want to talk a little bit about your development of our Center for Infectious Diseases. We have 6 centers of excellence at Houston Methodist. This is going to be our 7th. It's got obviously much development to happen. Talk some more about the recruitment that you're doing and some of those capabilities, and I guess fast forward 3 years from now, what does that center look like, and how uniquely does that position Houston Methodist?
Happy to address those points, Dr. Boom. So I think, first and foremost, importantly, we, in biomedical research, we are in an internationally competitive environment. So for the individuals who we want to join us in the Center for Infectious Diseases, they're going to be wanted by many different institutions. It's just like in the business world. It's international competition to get the best talent and to bring the best talent on board. So what we want to do right now, as I mentioned in my slides, is to bring on some of the key areas to fill our gaps. We really need an excellent human virologist. We need an excellent vaccinologist. And we really need to bring on board folks who can fill those gaps rapidly. In the future, as we move forward, those individuals who we bring on board to fill those gaps will be the leaders of new areas of research, just like, for example, Dr. Arias and Dr. Gallagher are doing. In the future, we will also have important areas to complement the existing expertise. There'll be educational activities as well. There will be the ability to get the word out in a much better fashion to the community about what we're doing and how we're doing it and the importance of what we're doing in the future. I think that very, very importantly, we can't lose focus, as Dr. Arias mentioned, we can't lose focus about the problems with the silent pandemic, this antibiotic resistant problem that will always be with us. So we've got to build this structure in the new center, all the while also increasing our ability to challenge and to fight against the stuff that we've already known is a problem, antibiotic resistance and so forth.
So you've done some work kind of in the genomic identification of some bugs, so to speak, right? Because I think clinically, oftentimes somebody comes in, now let's talk bacteria, and we know they're sick, we know they have pneumonia, we don't know what's causing it, so, usually the way we do that is we give people like this big, broad, big bazooka worth of antibiotics until we figure out what they've got, narrow it down. But what you work on is antibiotic resistance. So the more we put out big guns out there, the more we might push and select for resistance. You've done a lot of work to try and make that process a lot quicker so we can get to the refined treatment really quick. Can you talk about that a little bit?
Surely, happy to. So I think importantly in all of medicine, a key direction in all of medicine is going, not just from the standpoint of infectious diseases, but all areas of medicine, is into the precision areas. So what you're, I think, really alluding to, Dr. Boom, is to focus in a very precise fashion on the problem that we're infected with at any given time. To do that, we have to have rapid diagnostics and then rapid antibiotic resistance determination. So a lot of what we do in the clinical diagnostic microbiology area is focused on that. Rapid detection and then rapid identification of drug resistance are crucial. And frankly, Dr. Arias is very engaged in that arena as well. But this whole area of precision medicine is such a huge area now, and it's not just infectious diseases, but it's also in the area of precision antibodies as well. I mean, it's all focused on rapidly, in a fast fashion, the precise problem, addressing the problem with a precise therapeutic rapidly.
Right. So linking some of that together, it's precision's a great word. A lot of precision requires obviously very sophisticated techniques. You talked about, obviously we were one of the first institutions in the country that could stand up testing very, very early on, as a great example with COVID. What I was hearing from Dr. Gallagher is some of that sophistication, rapid throughput, it's AI, and it's the technologies you have. So it intrigues me, the platform you've created to look at a piece of a virus, basically, so you don't have to worry about infection. Because I think classically of kind of like high-end infectious disease research, you think about people in the white suits, and obviously we saw a lot of those images in COVID, right, and the different levels of containment. But now you're working with highly infectious organisms, but able to take a piece of them and actually do the same kind of work with no risk. Is that right?
That's correct. So we want to take these really dangerous pathogens and break them down to their protein components so that we don't have to grow them and wear bunny suits and have BSL-4 facilities. We can do this in a BSL-1, BSL-2 facility very, very easily, and we would like to keep it that way.
So talk BSL-1, 2, 4, talk about what that is.
So BSL-1 organisms are common laboratory organisms, something like in E. coli. BSL-2 has a slightly elevated risk associated with it. Some of those are mammalian cells, that sort of thing. Nothing too pathogenic. You can have some commensal bacteria or opportunistic pathogens will sometimes be BSL-2. They're not too bad. They're generally antibiotics or treatments to a BSL-2 organisms. We want to stay there and lower so that we don't risk our health in combating the disease, and we think we can do that.
Well, that's great. And I mean, that's just absolutely fascinating. So you talked also about pulling something out of the freezer. I mean, that's really, imagine COVID if we could have pulled out of the freezer a therapeutic, or could have pulled out of the freezer a vaccine that we already had that worked against all coronaviruses, for example. Right. How will you go about doing that? Because there's umpteen microorganisms, and some of the worst ones are the ones we don't even see coming sometimes, or that are jumping out of animals into humans, what we call a zoonotic infection and those kinds of things. How is it conceivable to do that? Is that practical?
I believe so. So there's a lot of surveillance sequencing going on in the world. There are a lot of spillover candidates. And what we want to do is take the major families of viruses, take and make countermeasures to those, and then see how broadly applicable that is to other family members. For example, SARS-CoV-2 was very closely related to SARS-CoV-1, which was also related to MERS. The reason we got a vaccine so quickly was our colleagues in Austin, Jason McClellan, had already pre-fusion stabilized MERS and SARS-CoV-1. So that was almost out of the
pre-fusion stabilized, define that for
so that's the spike protein adopts a pre-fusion conformation, and that's where you want antibodies to bind, because that will stop it from going post-fusion and entering the cell.
Got it.
So what we would like to do is figure out the tips and tricks on how to do that to different virus families, and then step through the most likely spillover candidates.
Great, fantastic. And let me ask, I'm reading now about another Ebola outbreak in Uganda. Is this one any different, or is this another, of the periodic outbreaks that we'll see there that can be contained because of the severity of illness and quick identification and the processes they have placed there?
Yeah, I think by and large, I hate to say it, but if you've seen one Ebola outbreak, you've seen one Ebola outbreak. They're all a little bit different. They have some variants that are important, but not to the extent of the level of genetic variation that we see now in SARS-CoV-2 and certainly in influenza and so forth. The ones that we're seeing now, the Ebola situation that's being described now, is by and large similar to the other Ebola outbreaks. I think, coming back, if I may, to one of Dr. Gallagher's topics, it is crucial, insofar as possible, to get into a predictive mode about pathogen evolution. It is really essential to use some of the, frankly, really cool high-end techniques that Dr. Gallagher's group is using to get at being able to, if you will, predict 3-4 steps down the road which variant we're going to be faced with, regardless of whether it's the SARS-CoV-2 or influenza and so forth. Now, that is an extremely, extremely challenging area that you have to bring together artificial intelligence, protein engineering, and so forth. And that's really part of our motivation in bringing Dr. Gallagher and his group on board. Because as he absolutely correctly alluded to, we want to be able, 5 years from now, when, just to pick a pathogen, Nipah virus, when it's introduced and comes on board, we want to be able to pull out of the freezer something that can be rapidly scaled up and used for therapeutics. Just like we'll do with the mRNA vaccines. That's such a powerful technique that will be deployed regardless of what the pathogen is in the future. And I'm not that adept at predicting the future, but I will predict that come October, which is right around the corner, there will be several Nobel Prizes given for some of the work that was done in the arena of mRNA vaccines and how successful those have been.
Obviously saved countless lives. I mean, just
millions
just
and
absolutely
millions.
Countless lives. Just amazing. Well, I'm going to wrap up with one last set of questions for you, Dr. Arias. I know your work is really fundamentally around antibiotic resistance, and your statistic of, I think, 4.95 million deaths is pretty sobering, and statistic of predictions by 2050 of more deaths than from cancer, and maybe some of that's cancer coming down, but I'm sure a lot of that is antimicrobial resistance going up. Can we win that battle? I mean, how do we think different? I mean, I think back to my medical school days, and if I look at a list of kind of the key antibiotics we use, they're not that different. I mean, obviously there's been some development, don't get me wrong, but not any major, big, huge new classes that suddenly have usurped old ones. I mean, how much work is there being done to really figure out the next way to kill these organisms that, obviously we will always deal with? So.
Yeah, this is a great question, and I think I want to highlight that the complexities of this problem. So in COVID, you have, let's say, one virus. I'm not trying to undermine the complexity. You have one virus, one target, maybe one protein. Here is a concoction of bacteria that we all live with. Actually, we carry all these bacteria in our gut, in our skin, and all that. So that's part of our evolution as human beings. That they're being sort of selected because of what we do in agriculture, what we do in the environment, what we do in clinical medicine, putting a lot of antibiotics in this. So it's a massive, complex program that involves all these areas. Secondly, the discovery of the gold era of antibiotics in the 70s, we sort of exhausted that pipeline. And that's one of the reasons, because discovering new antibiotics is difficult. In the past, pharmaceutical companies will go to the jungle and get a bit of earth and do some screening, and that's what big pharmaceutical companies actually were born on. Then the economics of trying to develop a drug where you use only for a few days, and then me as an infectious disease try not to use it, is not very palatable for pharmaceutical companies. So all the major pharmaceutical companies actually have pulled out of antibiotic research. And a small companies that have taken the challenge, even when the drug is approved, they have declared bankruptcy. So you need a really interesting partnership between the drug makers, the academicians and researchers to be able to pull that area, and the regulatory network that needs to be changed. So the UN has declared this as a major health problem. I know the Wellcome Trust and certain foundations are putting money there. There are initiatives, for example, now coming in Congress to try to change the way we pay for antibiotics, and discover that we in clinical practice take antibiotics for granted. We give what you say, ton of antibiotics because we have them, right? But we don't do the same with cancer drug or rheumatology drugs, and these drugs are actually cheap, and we think if we pay a little bit more, it's just too expensive. So we need to increase the value of these life-saving drugs, and I think until we have a model of that, this problem is going to be with us for a long time.
Well, that's a great close and a great explanation of really why we are making this infectious disease center of excellence. I think of the last century as kind of the century that we sort of took the upper hand against the bugs, and actually you look at why we had such an increase in life expectancy from 1900 to 2000. The biggest things all have to do with infections, starting with sanitation, of course, but then antibiotics, antivirals, vaccines, big time, et cetera. But I kind of think of this last 20 years as the bugs are really fighting back. So we've got to up our game. And so you've spoken about some of the challenges, and I know y'all are up to it, and really grateful that we have, really a rock star team here, and grateful to be with each of you here today. I'm going to wrap up with a quick presentation, quick update. This is probably the first of the 28. You're not even going to see me show a COVID slide because I had the real experts here today. Usually I'm giving you an update on where things are, but you've got a feel for that today for sure. So I'm going to give you a couple updates for Houston Methodist. We talk about our vision for the second century, 6 simple words: unparalleled safety, quality, service, and innovation, over and over and over and over again. And really it's what makes this institution tick. And along with that, we'll talk about, when you focus on those fundamentals, sometimes the awards follow. And so I did want to share with you some excitement we have around some awards, not entirely because the awards. We got to stop and celebrate, and really celebrate the people I'm here with today and all the wonderful people who work at Houston Methodist and what they accomplish, but really because of what they indicate about the fundamentals that we are focused on. The thing I look at the most closely is something called Vizient. Give you a little history of this. Some of you will have heard me talk about it before. Probably about 17 years ago, we worked together across all the academic institutions in the country to say, look, if we compete together on the fundamentals, all our patients will win and academic medical centers will thrive. And we began that process, and then over time we've done that and broadened it out to all of our hospitals. And what we've done is standardized data. When we call something this, so does Mass General. And when Mass General calls something that, so does Cedars-Sinai, et cetera, around the country. And so unlike many other kind of externally created surveys, this one really is very meaty, no reputation. It's all about the outcomes. What you see here is our institutional, across all our institutions over the last 7 years, which is how long they've been ranking all of the hospitals. What I do is I color code this for our team to keep it simple. If we're in the bottom half of those hospitals in that respective group, we get red. If we're in the second quartile, we're yellow. If we're in the top quartile, we're green. And if we are in the top 10%, we're blue. Then if you see 5 stars, that's actually Vizient saying, hey, you're statistically in this absolute top 5-star group that we've got. for the second year in a row now, all but one of our hospitals were in that absolute top Vizient group. The one that's not is Clear Lake, and actually still in the top 13% or so of hospitals. And interestingly, it's a small hospital, so it's in a little different group with a lot of like 10 or 20 bed hospitals. If it was in the next group, it would have ranked number 7. Really proud of how our hospitals have done. Maine Methodist, number 6, again, for the third year in a row now, out of over 100 who's who. This is all the major academic medical centers in the United States, minus a couple who don't participate, but pretty much everybody out there. So spectacular results there. Sugar Land's kind of in the next size down. The third time in 4 years now they've been number 2. They're like literally a couple decimal points away from being best in the country in their group. And then what you see is that next group down, we actually took out of well over 100 hospitals, number 1, 2, 3, and 6 in the country. And so incredibly proud. And we would have also had 7 if Clear Lake was in that group, in their group, still, as I said, about top 13%. Continue to drive. It's not about this, it's about the fundamentals, but it really shows that. When you look around the country and you look at the institutions and how many of their hospitals are there, we have 6 of our 7 there. The Mayo Clinic has 7 out of their 16 hospitals. We have profound respect always for Mayo Clinic as well. Intermountain Healthcare, which is based in Salt Lake City and has a kind of a flagship hospital, not quite as academic as most of the others on the list, but nonetheless in that group. And then lots of kind of small rural and other hospitals, very good system. They have 6 like we do out of, I think it's 24 hospitals. And so very proud of the consistency that we've been able to achieve. On an interesting note, we oftentimes don't really have a good way of sort of saying how we do it on the innovation front. It's always safety, quality, service, everything we were talking about. I joke that I think every used to be magazine that now has very little reason to exist with the internet, and certainly doesn't send out many paper magazines. They all figured out, let's get in some sort of ranking business, because that's what they're selling. We're selling. So Newsweek's been getting into the game as well. But one of the things they've done now for a couple of years is look at smart hospitals. I mean, basically looking at hospitals that apply many of the techniques clinically that we were talking about here today: artificial intelligence, big data, much other technology, et cetera, et cetera. Out of all of the world, we were ranked the number 10 hospital in the world. They have about 300 that they rank. So a nice pat on the back, really, for our innovation teams as we continue to drive and fulfill leading medicine. We got a Forbes magazine, yet another one of these magazines that is doing surveys, but looking at best employers. And of course, we can't be the best without the best people. You saw 3 examples of that here, but that's best people in every walk of everything that's helped with at Houston Methodist. And so very happy with that as well. And then yet another one, I think this one's Fortune. We were in their top 15 teaching hospitals. We were number 13 with this group as well. So it's nice to get some of those. And then a very specific one that I'm proud of. We have a very unique thing here, many of you are familiar with, called Center for Performing Arts Medicine. Really, at that junction point of our looking at faith and our looking at a holistic approach to caring for patients, and recognizing while the technology is critically important and you got to have that at the center of everything, it's a healing touch, and it's compassion, and it's a holistic approach, and the psychological approach and the spiritual approach and everything else to healing. That's really the whole package, and music is therapy, arts is therapy are a big part of that. And so the Texas the Texas Cultural Trust awarded us with their Medal of Arts Award this year. So kudos to all of our leadership in that area as well. Then just a couple items of some national visibility. Innovation continues to be a big push. Let me tell you, everybody's coming to us to see what we're doing. We're building, as I've talked about before, our Cypress Hospital as a smart hospital, as a hospital of the future. Roberta was just keynote speaker at a huge conference nationally last week, and really talking about how we are creating that smart hospital, how we are using technology, how we're using technology to reimagine care. We're in a major staffing crisis nationally, so we have to rethink how we care. We need to bring technology to bear and make clinicians' lives more meaningful, give them more frontline care with patients, and pull them away from the technology, have the technology be really in the background as a support, and all of that work that we're doing there. And then finally, almost simultaneously, actually, Vizient, which is the one I was referencing, actually asked me to be their keynote speaker at their annual conference last week, about 2,000 people there in attendance. really because of those kinds of results you saw. And what they asked me to do, and what I ended up doing, was talking about how our culture led to our ability then to thrive on the fundamentals, and how focusing on the fundamentals the awards are following, and you see those results. How our culture has driven the work that Roberta and team are doing, that she was featuring almost at the same time, around innovation. How our culture led our COVID response. And Dr. Musser's ears may have been burning because I joked with them that said he and I were on speed dial, and I think those were his dream conditions and conversations, because he'd be like, Mark, I need another $2 million piece of equipment. And I'd say, okay, like instantly. So I don't do that anymore, but he's got to work a little harder these days for it. But seriously, we were blessed to be able to drive that. And then really culminating in, how a culture of people in an institution as diverse as ours, in every shape of the word, including political thought, and how we approach the world, can have a common culture and can do a lot of good in the community together, and really a call to action for people in our community to say we can do that, all of us, together, because we do have a common culture as U.S. citizens. Sometimes we forget that, and differing opinions when I'm in a table with people in the middle of COVID and we're debating the science and we're debating what the right thing is to do, and one person thinks this and another thinks this, and we negotiate and we think and we get to the best solutions. That's when the best decisions happen. It's what I call the and we need more of that. And so it was a great chance to get us some visibility, really frankly a great chance to brag about people like I'm sitting here with, and all of the rest of the people at Houston Methodist. So with that, I'm going to wrap things up. I want to give you a little bit of information for the future. I want to thank, obviously, our presenters. I want to thank Dr. Miller, who's been answering your questions in this live stream chat. Our next town hall will focus on stroke prevention and treatments. It will be on Thursday, October 20th. So it's actually just the way timing's working out, only 3 weeks from today. So we'd love to see you then. If you want any information on what we're doing with infectious disease, by all means, reach out to us, and we'll be happy to share much more with you. Thank you, and have a great day.